Hello everyone, welcome to today's session. Today we are going to see class 12 biology chapter 5 molecular basis of inheritance. In that we are going to see the search for genetic material. So in the previous chapter we learned that genes control inheritance, but scientists had one important question. Which molecule actually carries hereditary information from one generation to the next? So in order to answer this question, several landmark experiments were performed. So today we are going to study four important discoveries. One is the Griffith transforming principle, and Avery, MacLeod and McCarty experiment, and Hershey Chase experiment, and also why DNA is preferred over RNA. Yes, let's start with the topic, children. Griffith's transforming principle. So the first clue about the genetic material came from Frederick Griffith in the year 1928. Yes, these years and all very important. Yes, so came in the year 1928. So he worked with a bacterium called Streptococcus pneumoniae, which causes pneumonia. So Griffith observed two different bacterial strains. One is the S strain, this is the smooth strain, and another one is the rough strain, that is the R strain. So let's see the characteristics of this smooth strain. See this smooth strain, the colonies are very smooth and shiny, and also they are covered by a polysaccharide capsule or coat, we can say, and this it protects from the immune system, and also it is virulent because it is causing diseases, and after the experiment the mice died because of the infection. When we take this R strain, they are rough colonies, and no capsule is visible there, and also no is non-virulent. Yeah, it's not causing any disease, and also the mice survived. So let's see the Griffith's experiment. So Griffith, he performed four experiments. You can able to see four experiments he performed. So in the experiment number one, he injected the live S bacteria into the mice. Yes, into the mice. So the mice died because the bacteria were virulent. In the experiment 2, he injected live R bacteria, that is the R strain, yeah, into the mice. So what happened? The mice remained healthy because R bacteria cannot cause disease. That's why. In experiment 3, he heated the S bacteria in order to kill them and injected only the dead bacteria. So the mice survived because dead bacteria cannot infect. In the experiment 4, Griffith, he mixed this heat-killed S bacteria and live R bacteria and injected into the mice. So unexpectedly what happened? The mice died. So when he examined the dead mice, he found the living S bacteria. So you may have a question, how this happened? Yeah, the dead S bacteria, it somehow transferred something to the live R bacteria. That unknown substance changed R bacteria into disease causing S bacteria. So this phenomenon is called transformation. This phenomenon is called as the transformation. And the unknown substance, it came to know as the transforming principle. And Griffith, he concluded that the hereditary information had been transferred. So he could not identify whether the transforming principle for DNA, protein or RNA. Yes. So let's move on to the next one. Biochemical characterization of the transforming principle. So after the Griffith discovery, scientists wanted to identify the chemical nature of the transforming principle. So what happened between the year 1933 to 1944, Oswald Avery, Colin MacLeod and McCarty. So they performed experiments. And what was their question? Was the transforming principle a protein, RNA or DNA? So what was their methodology? They purify different molecules. They purify different molecules from heat-killed S bacteria, and they separated the protein. Yes, what they separated? They separated the protein, RNA, and the DNA. Then mixed each with the live R bacteria. Then they performed enzyme experiments. So first enzyme they took were protease. The protease, it destroys the protein. So after adding protease, transformation still occurred, and it is proved that the protein is not the transforming principle. And the next one is the RNAs. RNAs destroys RNA. So transformation still occurred after that. So hence it is proved that RNA is not responsible. And next one, DNAs. DNAs destroys DNA. Now transformation stopped completely. The R bacteria never became S bacteria, and this proved that DNA is essential for transformation. And he concluded that only DNA can transfer hereditary information. Therefore, DNA is the transforming principle. But still, many scientists believe that proteins might be hereditary material because proteins appeared more complex. So here are some exam tips. Generally, students they used to confuse between this DNA with the DNAs. See, DNA is the hereditary molecule. DNAs is an enzyme that cuts DNA. Let's move on to the next one. Hershey Chase experiment. So although Avery's experiment strongly supported DNA, many scientists still had doubts. Finally, Alfred Hershey and Martha Chase, they provided the final proof. They used viruses called bacteriophages. You can able to see the bacteriophage picture over here. Yeah, these are bacteriophages. So what is a bacteriophage? Bacteriophage is a virus that infects bacteria, and also it consists of a protein coat and DNA inside. And when it affects infects bacteria, only the hereditary material enters. The question was, does DNA enter or does protein enter? Then in order to find this one, they did one experiment with the help of some radioactive labeling. So scientists used radioactive isotopes. The radioactive isotopes used here are phosphorus, that is a P32, and sulfur S35. And this DNA contains phosphorus. Proteins do not. Therefore, DNA becomes radioactive with the help of phosphorus. And the radioactive sulfur, proteins contain sulfur, DNA does not. Therefore, the protein becomes a radioactive. So, we are going to see the experimental procedure with the help of the diagram given here. I'll explain you each and every diagram. Let's move on to the diagram. So, let's take this one. So, in the first one, the bacteriophage with the radioactive protein. You can able to see what radioactive protein is here. That is a S35, right? Attaches to the E. coli bacterium. E. coli bacterium is nothing but Escherichia coli. Yes. So it injects its DNA into the bacterial cell. It injects its DNA into the bacterial cell. The protein code remains outside. You can able to see the protein code. It is remaining outside. And next thing is blending. So the mixture is placed in a blender. Then the blender shakes off the empty. You can able to see it is going off the empty viral protein codes attached to the bacteria, and only bacterial cells remain intact. Next process is the centrifugation process. During centrifugation, this heavy bacteria, they settle at the bottom in the form of pellets, and the light viral codes, they remain in the upper liquid. Yes, that is the supernatant. Then scientists checked whether the where the radioactivity was present. So they observed the bacteria showed no radioactivity. The supernatant contained the radioactive sulfur. Yes, the supernatant contained the radioactive sulfur. And what was the conclusion? Protein never entered the bacterial cell. It remained outside. Therefore, protein is not the genetic material. So let's see this side. So now another bacteriophage is used here. So its DNA is labeled with the radioactive phosphorus. So the virus injects its DNA into the bacterial cell. Then the blending process, the blender removes the empty protein coat. The DNA remains inside the bacteria. Again, centrifugation is done. The bacterial cells settle at the bottom, and scientists check the radioactivity. And what they observe, the bacterial cells contained radioactive phosphorus, and the supernatant showed no radioactivity. And what was the conclusion of this experiment? DNA entered the bacterial cell and directed the production of new viruses. Therefore, DNA is the genetic material. So next we are going to see properties of genetic material DNA versus RNA. So after proving DNA is hereditary material, scientists asked another question. If both DNA and RNA are nucleic acid, then why DNA the main genetic material in almost all organisms? This question arises in our mind also, right? So the answer lies in the properties which is required for a genetic material. So let's see the properties of a good genetic material. The first one is the replicate. So you should make exact copies of itself. Both DNA and RNA can replicate. And next one is the stability. Be chemically and structurally stable. So the molecule should remain unchanged over generation. Let's talk about the DNA first. DNA is more stable because it is double stranded. Next, it is having complementary base pairing, and also they are less chemically reactive and it contains thymine. And RNA, they are unstable because they are single stranded and they have a reactive OH group at the 2' end. And also they are easily degraded and it acts as a catalyst. And next one is it allows low changes for evolution. Changes immediately comes to your mind, mutation. So mutation creates variations needed for evolution. DNA mutates slowly, RNA mutates rapidly. So this explains why RNA viruses evolve quickly. Examples we can give like a tobacco mosaic virus, yeah, TMV, QB bacteriophage. And next one is the express as Mendelian characters in order to express the information. See, genes should produce proteins. RNA directly participates in protein synthesis. And DNA, what it does, it first transfers the information to RNA. Then the RNA synthesizes the protein. And why DNA is preferred? See, DNA is preferred because it is more stable. Yes, it is double standard. And also they are very less reactive and better storage molecule and easily repaired using the complementary strands and thymine. It provides additional stability, right? And why RNA is important? It carries the genetic information. What it does, sir? It carries the genetic information and transfers the information from DNA and also helps to synthesize the protein and also it evolves rapidly. Yes. So let's move on to the next part. Let's have a quick revision, the search for genetic material. So Griffith discovered transformation but did not identify the molecule. Avery, MacLeod and McCarty, they showed DNA is the transforming principle using DNAs. Hershey and Chase, they proved DNA enters bacteria while protein remains outside. DNA is more stable than RNA and is therefore the primary genetic material. And RNA acts as a genetic material only in some viruses and it plays a major role in protein synthesis. So here I have given you a short thing of all the important things. Yes, just see S strain, how the characteristics of the S strain and also R strain characteristics and notice the transforming principle, Visa, yeah, and Avery et al. Show DNA is the transforming principle. DNA's test, only DNA stops transformation, that is to DNA. And bacteriophage, virus that infects bacteria, and these are the phosphorus labeling the DNA and the sulfur labels the protein. And Hershey Chase, they proved DNA that is not protein enters the bacteria. Stability, DNA is more stable and storage of information. RNA genome, it mutates faster and is better for transmission. So we have come to the conclusion part. Watch the full video. Yes, we'll see you in the next video. Thank you.